Your Excellencies, distinguished participants, I'm Victor Dzau, the President of the U.S. National Academy of Medicine. And what I'd like today is to give you an overview about the signs of COVID-19. You heard how devastating this pandemic is and how it's affecting lives lost, human suffering, communities, people's lives, and hurts the economy. It's truly affecting everyone. We've done a lot in this area, but there's still a lot need to be done. We know something about the virus, but still a lot that need to be learned. And next slide, please. So I'm going to try to touch on in 10 minutes this scope of things. What do we know about the virus? What's the state of the pandemic globally? What are the effectiveness of containment mitigation strategy, testing, treatment, vaccines, and of course, role of technology, lessons learned, and how are we prepared for the future? Next slide. As of this morning, we can see there about globally about 4.5 million cases, about 300,000 deaths. The United States turns out now to be the epicenter with the largest number of cases, 1.4 million, and about 85,000 deaths. Next slide. And so what do we know about the virus? This is a new virus of the coronal family, which is called SARS-CoV-2. It's highly contagious, more contagious than Ebola, SARS, and others, and results in more severe illness than H1N1 and all the others. This family of coronavirus, there's only 6 other members that can infect human. As a matter of fact, 4 of which have been around for more than a century, and they cause a third of the common colds that we all get. Now, SARS-CoV-2 affects both upper and respiratory tracts, and the lower respiratory tract leads to pneumonia, and attacks other organs such as kidney, heart, can lead to marked inflammation of many tissues, including the blood vessel, and associated with the childhood vasculitis syndrome, which looks like Kawasaki. And there's also increased coagulation, thereby increased thromboembolism. So you can see this virus is a very aggressive virus. In fact, when you have the disease, you can also end up with what they call a cytokine storm, which is devastating and frequently is cause of the last stages of one's life. The virus you see in this picture is a ball and spikes. The spikes are known as spike protein because you have the envelope protein, membrane, and nucleic capsid. But importantly, the spike protein is the one that binds, as you can see, to a receptor on mammalian, on human cell surface called ACE2. And by entering with ACE2, it can enter the cell in the endosomal pathway, and then with low pH, it can be released into the cell, causing infection. Another protein which is very important is TPM-RSS2, which primes the spike protein to be able to bind to ACE2. Because of distribution of ACE2 broadly, that's why this virus is able to affect many different tissues. And unlike SARS, for example, or MERS, which binds to a peptide called DPP4, which is distributed less aggressive in many tissues. So that's at least one explanation of why this virus is so aggressive. Next slide, please. So what do we know about transmissibility of the virus, the mode of transmission, how does it spread, what's the incubation period, does it live on surfaces, and so on. So what we do know is that the incubation period of this virus is about 1 to 14 days. And as many have heard, you can transmit it during asymptomatic period, which means that the most infectious period is thought to be actually 1 to 3 days before symptoms. And it can persist for the first 7 days after symptoms begin. And many people can retain infections for a much longer period of time. So we do know it's mainly airborne, addition to droplets as well. So aerosolization and of course other droplets is one of the reasons why we are doing 6 feet or 2-meter distancing to reduce the spread of the virus through airborne or even droplets. It does remain on surfaces. Evidence suggests that it can remain viable for hours to days on surfaces, variety of materials. So yes, at least in our country, the virus is with us, despite our different strategies. One would expect that if we do open up and not careful, this virus will infect again. So the virus is not going away. And of course, in the African countries in the south, we heard from the ambassador of Ghana and others, when the colder days begin to occur in the months, July onwards, you may see a larger spread of this virus later on. So I think we are certainly expecting this to be around for a while. We hope it will not become endemic. The virus is mutating. There's lots of studies showing that the mutation can change its transmissibility. And of course, the question about if you had the virus, are you immune? And I'll talk about that in a few minutes. Next slide. So as many of you know, public health interventions are very important. And different countries have used different strategies. Containment, which is used very early, identifying the case, and then having contact tracing, quarantine, isolation has been very effective in China, South Korea, and Taiwan. China, with a population of 1.3 billion, they have today 82,000 cases, as you see the aggressive close down of Wuhan and Hubei province. As is South Korea, with 11,000 cases, and Taiwan with only 440 cases. The countries like US, UK, and Europe came late into the equation, so the strategy became one of mitigation, which is simply trying to contain the virus through non-pharmacologic interventions like social distancing, hand washing, many others, but in so doing is trying to reduce the surge and create a strain of the health system. And because the health system simply doesn't have the capacity to deal with so many cases. So now, as we're living now in the United States into our third month, we're seeing possibly a flattening of the cases, but still we know the cases are still among us and continue to spread. Next slide. So testing becomes really important. The ability to diagnose, the ability to track, to survey, and others. So molecular testing such as reverse transcriptase PCR, or more recently, the more novel CRISPR testing, allows you to know the current set of infection. Antibody testing or serologic testing allows you to know whether you have developed antibody and implies immunity, but right now we are not absolutely sure if you're able to detect antibodies in someone who's post-infectious, whether these antibodies are neutralizing and really gives you immunity. That work is still ongoing. And certainly we heard of cases of people getting infected again, and these are rare cases, but I think there's still a lot of work need to be done to know whether simply having antibody means you're immune. There's a need for more rapid testing because PCR, too difficult. Certainly in the early days, you need to do oropharyngeal swab, then put in the culture, test it with culture media, send it to a laboratory, because people are trying to develop point of care testing, but with antigen, possibly immediately measuring the antigen, you could be able to detect right away. I should quickly point out to you, antibody testing, there's still a lot of false positive and false negative. The specificity and sensitivity is still quite poor. And then antigen testing is still very much in the work. But we all know that testing is central to screening, diagnosis, monitoring. I believe that the importance is to use testing very early, very, very early, to identify the cases, do the contact tracing, quarantine. That's the most effective strategy. In our country, where now it's spread so far spread, we need to know and to use it to not only to find the cases, but survey and also to create strategy of pharmacologic and non-pharmacologic interventions. Next slide. This leads me to talk about treatment. There are now a number of possibilities. Many of you know about the antiviral remdesivir, which recently has at least early report a randomized controlled trial that seemed to reduce the severity of the disease, but not reduce mortality. It reduces the hospitalization of the patients when you receive it, when you're hospitalized. There are more broader antivirals, broad-spectrum antivirals, which are not proven yet to be effective. Many people are looking at what happened if you had the infection and you have large number of immunoglobulin that can neutralize the virus, so the convalescent plasma or using hyperimmune immunoglobulin. That is an active area of work in our country, in China, and elsewhere. There are areas where drugs which are repurposed, such as hydroxychloroquine and others, but as you know, this has been very controversial, and there's no definitive data that hydroxychloroquine works. I mentioned earlier about the cytokine storm, high levels of IL-6 and others. So there are, in fact, immunomodulators which are used, at least experimentally, to do this. There's also monoclonal antibodies and others. Suffice to say that therapeutic armamentarium, at least as been proven to work, is still very small, but lots of clinical trials, large number, over 1,000 clinical trials globally in these areas. Next slide. Vaccine. The good news is that because of technology development, the platforms now are very effective. So you can actually create a vaccine fairly quickly, at least a vaccine candidate. For example, nucleic acid based like RNA and DNA can produce, in fact, a vaccine candidate very quickly. In this case, matter of 40 days in our country. You can also use recombinant protein, which is more traditional, peptide-based, nanoparticle, of course, live attenuated, inactivated, or using viral vector like adenovirus. Many have heard, of course, the work done is, for example, in Oxford, Cambridge, that is using a part of genetic material from the virus, which encodes for the spike protein, put in an adenovirus, and now they are, in fact, moving into phase 1, phase 2. Altogether, there are 174 vaccine candidates, next slide, in 10 clinical trials. And as you can see, some of the candidates are already in phase 2 and certainly in phase 1. One would expect some really results of efficacy and safety by the end of this year, maybe even earlier. But of course, definitive result, we may have to go into next year. But part of the vaccine issue is not only getting the vaccine that works, but having supply and having distribution, equitable distribution. Next slide. So this is a slide that shows you an example of some of the vaccine that is ongoing from the companies to different phases. I won't have time to go over this. Next slide. Okay, so I want to talk about solidarity. And with all of you ambassadors and people from different countries, we need to work together. Because even though we have multiple trials on vaccines, and one or more could be positive, we need to be able to assure every country to have access, to have that distribution, to have equitable distribution. So in that context, we need to work together and every country together towards this solution, because no country should be left behind. The problem we're always worried about is those who have economic power would buy the vaccine and get them much earlier, and the poorer countries will not have access to them. So with that, I want to tell you about a really important initiative that started off with a pledge. I'm a member of GPMB, which is the Global Preparedness Monitoring Board, which, as you know, was created as a result of the UN High Commission after Ebola to say there should be a board to oversee the monitoring of preparedness. This is chaired by Bro Grundlund. Grundlund, as you know, previous Prime Minister of Norway and DG of WHO, and a C, who is in fact the president of the International Federation of Red Cross. We, GPMB, said in response to this pandemic that we should raise a lot of money to put into vaccines, treatment, diagnostic. So we call for an $8 billion pledge, which was actually hosted by the leadership of President von der Leyen and the European Commission. As you can see, this is the pledge which happened 10 days ago. And the good news, within one day, where we asked for $8 billion US dollars, we exceeded 8 billion. For 7.6 billion euros, we got 7.4. With good exchange, the US dollars are high, we end up passing 8 billion. And I had the privilege of being one of the speakers, along with many of your heads of state. And this was very successful. This was a catalyst, next slide, to bring together call to action with the global leaders, government leaders, WHO, among with all the other stakeholders, CEPI, Gavi, Global Fund, Wellcome Trust, Gates Foundation, to create a unified approach now called Access to COVID Tools Accelerator. This accelerator will enable us to do how to assure rapid R&D, manufacturing, to procurement, to equitable distribution to all countries. And this consists of 3 separate pillars: vaccine, diagnostic, and therapeutics. And we're now working actively in creating end-to-end strategy to be assured that every country have access to the novel innovation that's going to stop this pandemic or to treat it altogether. Next slide. So I've given you a very rapid discussion of the science and epidemiology and treatment of this devastating pandemic. The question, of course, is technology. This is a big part of your conversation today. We, of course, recognize that digital technology using Bluetooth, GPS, and iPhone, many places now, including our country, are using technology to survey, to look at social distancing, and to be able to contact trace. The use of this technology will move much faster than the manual way of trying to track every case. So hopefully this will break through for countries that still haven't quite seen that many cases yet. And of course, for the future, a very important part. Having learned from all this experience, I would say to you, national academies have published many reports, including after Ebola, about lessons learned, what needs to be done. And of course, we're also slow, and because lessons learned are lessons forgotten. We must therefore work together to be well prepared for the future in the post-COVID world, and I look forward to working with all of you. Thank you very much.